CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adopted into a powerful tool for genomic research. There are two components to this system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. Cas9 has to implicate and bind to a common sequence in the genome called a POM. Once the POM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nucleus remains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduce mutations that disable the gene. This makes the CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of the Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene or introduce a stop codon at a specific place. Who are the persons behind it? CRISPR was discovered by Dr. Jennifer Doudna in UC Berkeley, seen on right in the image above, and Dr. Emmanuel Charpentier, Max Planck Unit for the Science of Pathogens, Berlin on the left side. Their groundbreaking paper revealing that the CRISPR-Cas9 bacterial immune system could be repurposed as a gene editing tool was published in the journal Science in 2012. It wasn't until 2020, well after it had been adopted in labs around the world, that Doudna and Charpentier won the Nobel Prize in Chemistry for their discovery, becoming the first all-female team to do so. Other significant contributors include Feng Zhang at the Broad Institute of MIT and Harvard in Cambridge, Massachusetts, who pioneered the use of CRISPR in eukaryotic cells and discovered novel Cas variants. George Sturge at the Harvard Medical School in Boston, Massachusetts, who was among the first to demonstrate its use in human cells, and biochemist Virginijus Siksins at Vilnius University in Lithuania, who independently discovered the ability of CRISPR to edit genes in other organisms. For more information on these early pioneers, you can check out our blog about prominent CRISPR scientists.
The usefulness of this developed technology and its sustainability. CRISPR spacer sequences are transcribed into short RNA sequences, CRISPR RNAs or CRRNAs, capable of guiding the system to matching sequences of DNA. When the target DNA is found, Cas9, one of the enzymes produced by the CRISPR system, binds to the DNA and cuts it, shutting the targeted gene out. Using modified version of Cas9, researchers can activate gene expression instead of cutting the DNA. These techniques allows the researchers to study the gene's functions, and research also suggests that CRISPR-Cas9 can be used to target and modify typos of the 3 billion letter sequence of the human genome in an effort to treat genetic disease. Clustered regularly interspaced short palindromic repeats or CRISPR is the most advanced gene editing technology yet. It works by harnessing the natural mechanism of the immune systems of bacterium cells of invading viruses, which is then able to cut out infected DNA strands. This cutting of DNA is what has the power to potentially transform the way we treat disease. By modifying genes, some of the biggest threats to our health like cancers and HIV could potentially be overcome in a matter of years.